Here I will be discussing about 5' ending capping, 3' poly-A tail, RNA splicing, alternate splicing, mRNA, tRNA processing, ribosomal RNA. What is post-transcription modification? The primary transcript is generally made up of RNA polymerase, which normally undergo further alteration, which is known as post-transcription modification. Generally, the RNA which is there before the post-transcription modification is the premature RNA. So after the transcription, after the post-transcription modification happens, then this RNA is known as mature RNA and can further can proceed to the translation process in the cytosol. So in prokaryotes, mRNA transcribes directly from DNA template and can used immediately in protein synthesis. And in eukaryotes, primary transcript must be processed to produce the mRNA, which is the active form, and this process occurs in nucleus. In post-transcription modification, the three processes happens, that is 5' capping, 3' poly-A tail addition, and RNA splicing, where the introns are removed from the immature mRNA and exons are joined together. So firstly, 5' end capping. In 5' end capping is modified by removing of the phosphate. Here it is modified by removing of the phosphate group from the triphosphate functional group, which is done by the enzyme triphosphatase. And after the removing of the phosphate group, then the guanine transferase, with the help of guanine transferase enzyme, guanine group is added to the mRNA. And after this addition of the guanine transferase to the mRNA, there happens the addition of 7-methyl transferase to the guanine transferase mRNA, and thus there is an ending of the 5' capping. So here is a picture from where we can see all the steps happening in 5' capping. And now I will be talking about the poly-A addition. In poly-A addition, the guanine this process of poly-A addition happens when the RNA is actually attached to the DNA. So, in this poly-A addition processes, there actually occurs two processes. First, the cleavage of the RNA, which is actually attached to the DNA. So here we require the cleavage factors. So this cleavage factors are actually the two factors which are made. The first one is cleavage and polyadhesion factors, that is CPSF, and the next is CSTF, cleavage stimulating factor F. Actually, these two attach to the mRNA, which is actually joined to the DNA. So, these two factors will attach to the mRNA from where the cleavage will happen, and by this the cleavage happens and the mRNA gets detached from the DNA, which is formed. And the next step, which is the addition of the poly-A tail. This addition of the poly-A tail actually occurs in that RNA which is actually formed from the DNA. So this happens with the help of poly-A adenylated polymerase enzyme. By this enzyme, the poly-A addition happens, and again the continuous poly-A addition can also be helped by the use of another enzyme, that is PAB2, that is poly-A binding protein 2. With the help of this enzyme, like continuous around 200 to 250 nucleotide of poly-A tail joins to the premature mRNA. By this, the mRNA gets poly-A addition, which can actually save, we can actually make the mRNA to stabilize, and it provides resistance to ribonuclease degradation and helps in transport of mRNA from nuclear membrane to transport into the cytoplasm for the further process of translation. So the next process which I am going to discuss is mRNA splicing. Here in actually the in mRNA splicing, uh, the mRNA which contains two the premature mRNA actually contains two types of pre-mRNA, that is exons and introns. In this process, we will be cutting all introns out of the mRNA so that we can be rejoining the exons present and thus making a mature RNA. So the 5' splice site is GU and 3' splice site is AG and the branch point is A. So here what happens, mRNA splicing occurs on heterozygous nuclear RNA. Splicing is mediated by large RNA RNP complex spliceosome. It contains a specific set of base uracil rich snRNPs associated with proteins. snRNPs helps in recognizing 5' end and bringing sites together and for cleavage. Spliceosome complex. The complex actually consists of five spliceosome units, which are U1, U2, U4, U5 and U6. U1 actually binds to the 5' site recognition. U2 actually joins to the A, which is known as a branch site, and U4 forms base pair complex with actually binding to U6 also, and U5 binds to the 3' junction binding of U4 and U6 complex. The spliceosome recognizes introns starting with 5' UG and ending in AG or 3'. In the mechanism of spliceosome, firstly, the U1 complex which comes and binds to the 5' splice site, that is UG, and then the U2 which binds to the branch point A. Here actually in U2, like in A site, the OH group is present, which actually helps in attachment of A zone to the UG zone, so that the bending happens, and this is done with the help of oxygen atom. So after that, uh, three complex like U4, U5 and U6 comes and attach to the premature mRNA. So after the attachment of U5, U4 and U6, the U1 and U4 protein gets out of the mRNA, and thus U5, U6 and U2 complex helps in cleavage of the intron and binding of the two exons. So thus making a lariat intron, a lariat structure which is also known as lariat intron. So, the pre-mRNA are spliced in several different ways, following a single gene to code for multiple proteins. The generation of different mature mRNA from a particular type of gene transcript can occur by varying the use of 5' and 3' splice sites. So here comes the mature RNA. So in mature RNA, it contains 5' cap, 5' untranslated region, start codon, coding sequence, stop codon, 5' and 3' untranslated region, and poly A tail. Here now the processing of tRNA comes. tRNA is also a form of RNA. So in this processing of tRNA, which occurs by the removal of ladder sequence and Taylor sequence, replacement of nucleotides, and in modification of certain bases, we have replacement of U residues at the 3' end of pre-tRNA with a CCA sequence, and then happens the addition of methyl and isopentyl groups to the heterocyclic rings of purine bases, and then happens the methylation of the 2' OH group in the ribose of any residues, and lastly the accretion of the introns. This is the tRNA processing and maturation. Here we can see all the processes, that is the process of attachment of uracil, then CCA complex formation and maturation, addition of methyl groups, and several processes happening. Next comes the ribosomal RNA. Ribosomal RNA in the cell contains 80% of the rRNA, serves to release mRNA from DNA, act as ribozymes in protein synthesis, relatively rich in GC-rich nucleotides. Ribosomes comes into play in eukaryotes and prokaryotes. In eukaryotes, it is 80 S subunit ribosomes, and in prokaryotes, that is 70 S. So in prokaryotes, if the ribosome is divided into 50S subunit, that is a larger subunit, and 30S subunit, that is a smaller subunit, which contains proteins respectively as 31 and 21 proteins. And in eukaryotes, we have 60S subunit, that is a larger subunit, and the 40S subunit, that is a smaller subunit. And with this, they contain 50 proteins and 33 proteins respectively. In this processing of ribosome, the transcription occurs, and thus the cleavage occurs, the further trimming, where 23S RNA is formed along with 5S rRNA, and tRNA is also formed. Along with the this is one of the form of rRNA ribosome, and another is the formation of 16S rRNA along with tRNA. The processing of ribosome actually occurs in 45S molecules occurs inside nucleolus. 45S molecules tightly associate with proteins for R and P proteins. First cleavage occurs at site 1 and removes 5' terminal ladder sequence, producing 41S intermediate and 18S. The second cleavage occurs actually at 41 as intermediate, at site 3' generates 32 as intermediate. The final cleavage, that is a separation of 32 as intermediate into 20s8 and 5.8s. The processed rRNA contains, as I said, 28s, 5.8s, and 18s intermediate. These are my reference, and thank you.